So this means that we now know that Parsortix is not only the only FDA-cleared system, the US regulatory authority, for recovering cancer cells for subsequent analysis, but it is also the only system that can look at these large CTC clusters. And in addition to that, it's by far the best system for providing cancer cells undamaged, alive, and intact for molecular analysis.
Hello, you're watching Proactive. I'm joined by Angle Chief Executive Andrew Newland. Andrew, very good to speak with you again. So can you tell us what's been announced today?
Good to see you. Yes, well, there's more pivotal research fundamentally enabled by Parsortix. So we're very proud to see some incredible findings from the Marchetti lab. This is the laboratory at the University of New Mexico Comprehensive Cancer Center run by Professor Dario Marchetti. And they've got some really impactful new data, which has been fundamentally enabled by the Parsortix system.
What is the research, Andrew?
Well, the research is about cancer metastasis. So cancer metastasis is how cancer spreads. And that's something that the Parsortix system is uniquely capable of supporting because it investigates cancer cells in the blood, and that's how cancer spreads. So more than 90% of patients who die from cancer die from the spreading to other organs. And Professor Marchetti's laboratory has used the Parsortix system to investigate circulating tumor cell clusters. You'll recall that recently we announced another group in Switzerland had found some groundbreaking work on CTC clusters. So what this is clumps of cancer cells, and the Marchetti lab has found for the first time that a variety of different things, but the targeting of those circulating tumor cells between one location and another. So they don't just randomly end up in a different location. They actually have signaling on the circulating tumor cells, which is caused by B cells. Now that's a bit of a complicated, the whole area is incredibly complicated. And we made sure that our announcement was reviewed literally word by word by Professor Marchetti to make sure that it was absolutely accurate. But I'll try and put it in simple terms. What they found, they developed two mouse models. So one mouse model which had an immune deficient system. So they added human cancer, melanoma cancer, to mice with no immune system and watched how the cancer progressed. They created a second mouse model, which was a humanized immune system. So these mice did have an immune system, but it was a human one, not a mouse one. I know that sounds a bit strange. And they watched how the cancer progressed in the humanized mouse model, and then compared the two. And obviously, you'd expect that the model had an immune system would contain the cancer and be much slower in terms of spreading. But actually, they found exactly the opposite. The cancer spread faster where there was a human immune system. And they were able, using Parsortix, to recover the cancer cells that were spreading and look at them very carefully. And they found that these circulating tumor cell clusters, clumps of cancer cells, had actually attached themselves to B cells, which are immune cells in the mouse model. And these so-called CTC B cell clusters, which this group has investigated for the first time, promoted the spreading of the cancer and made it go faster. And it also promoted the directional location of the spreading as well. That's two major findings which were not known of. And then a third one was that they looked at melanoma patients who had got primaries but not spread. And they found ones which had got progressive disease and then the cancer was spreading. And they found that before it spread, they could find large numbers of B cell CTC clusters in those cancer patients' blood. So that might be a way of determining who's going to have a progressive melanoma cancer versus a non-progressive cancer. And they surprisingly found that when it had progressed, the numbers of the B cell CTC clusters reduced. So there's lots of very complicated things there. But the read across is that this is a whole new area of investigation and understanding of cancer development, which is enabled by Parsortix.
Andrew, what are the implications for Angle?
Well, we think this is very important because it's yet another example where Parsortix is opening up completely new understandings of how cancer spreads. And obviously, in terms of the read across to Angle, that means that there's intense interest from many other researchers to investigate similarly. And this work can only be done with the Parsortix system. So that read across is very important. We also believe that pharmaceutical companies will begin to investigate whether they can come up with ways to block this metastatic spread. Different ways from the way that we talked about with the Aceto lab last week, actually. And so still more interest in driving the adoption of Parsortix to not only understand, but to come up with solutions to prevent and suppress cancer metastasis. The Professor Marchetti was very, very clear in his statements about the use of Parsortix, that it's the only system that is capable to get these large CTC clusters. So this means that we now know that Parsortix is not only the only FDA-cleared system, the US regulatory authority, for recovering cancer cells for subsequent analysis, but it is also the only system that can look at these large CTC clusters. And in addition to that, it's by far the best system for providing cancer cells undamaged, alive, and intact for molecular analysis. So for all those reasons, we see the Parsortix system becoming really now validated as the best sample for this kind of liquid biopsy analysis.
Andrew, so what's next for Angle then?
Well, we've got a very busy day today, actually. There's lots of things going on, but later today at 4 o'clock, in fact, there is the Illumina webinar, where Illumina, following the excellent results on DNA dual analysis using Parsortix, have allocated their webinar for the AACR conference to Angle to present our DNA dual analysis results. So that's an excitement that's coming later today, and there's been a lot of commercial work behind that. So we and Illumina have each reached out to literally thousands of potential interested parties, so cancer researchers, pharmaceutical companies, and others who have a very active interest in this sort of adoption. And we're hopeful that this webinar is going to be the beginning of major working together with Angle and Illumina, and the opportunity for Illumina customers. So these are customers who spent hundreds of thousands, if not millions of pounds on Illumina equipment, the leading sequencing platform, to now look at acquiring Parsortix, because that way they can get the best possible sample for analysis on these sequencing systems. So it's an important day for Angle, and we expect to see a lot of ongoing commercial activity as a result.
Well, Andrew, I hope you'll keep us posted on any new developments. Thank you very much for speaking with us today.
Thank you.
That's Andrew Newland, the Chief Executive of Angle.